Welcome everyone to the British Society for Gene and Cell Therapy Public and Patient Engagement Seminar Series. In this seminar, our aim is to share the latest development in this field. And thank you very much for joining us today. Before we begin, I'll go over a few housekeeping points. We will take your question during the talk. You can put your question in the live chat or in the comments. And to use the live chat, you need to sign in YouTube account or Google account. And then please note, these messages are not private. And also, please include your name and affiliation when you ask a question. And we will ask the speaker your question and at the end of the talk. By the way, I'm Özge, and today I'm joined by Raj, Kwan, and Sonam. We host patient and public engagement series together. Also, we are all researchers at University College of London. We are very excited to host Professor Paul Dalby today. To briefly introduce him, Professor Dalby is a co-director of the EPSRC Future Targeted Healthcare Manufacturing Hub and a deputy head of the Department of Biochemical Engineering at University College London. His research focuses on improving the design and formulation of therapeutic proteins and gene therapy vectors. His work aims to enhance their manufacturing, stability, delivery, and function. He has pioneered smart directed evolution approach to accelerate evolution in the lab. More recently, his work has used molecular dynamic simulation and computational protein design to improve protein and viral vector stability and function. So without any further delay, I'll hand over to Paul.
Great, thank you. And I can move on. Yeah, so I'm going to cover what is really a number of different strategies for capsid engineering and taking into account lots of work that's already going on elsewhere, but hopefully a slightly different take focused more on the manufacturability side of capsids. So yeah, I've already had the introduction that I co-direct the Future Targeted Healthcare Manufacturing Hub. So some of the work has come out of this endeavor, and this is really a pre-competitive research piece in collaboration with many companies aimed at improving manufacturing of biologics and gene therapies. So probably I don't need to do much introduction on AAV as a gene therapy vector. Many of you are familiar with it, but I'm going to focus primarily on the capsid, which is icosahedral. It's formed of 60 copies of essentially a single protein, but there are actually slightly different forms, VP1, 2, and 3, of that capsid protein at a ratio of 1 to 1 to 10. But really, they all contain the VP3. It's just that the VP2 and 1 are extra bits on the inside of the capsid. So really, all the focus is on VP3 and engineering that part of it. Clearly, it's used to deliver a gene of interest, usually within a single-stranded DNA piece, and it's being exploited in many areas for gene replacement, silencing, editing, and even gene addition. So yeah, it seems like a very promising vector, and there's a few on the market. This just shows you the sort of makeup of the genome, and so this is the DNA that's encoded, encodes the capsid. And this is typically how it's displayed between ITRs, but we now put this on another plasmid and put the gene of interest in there, which makes it into the gene therapy vector. I've already mentioned these three different VP3s, which VP1, VP2, VP3, VP proteins that are kind of expressed from the same gene, but with different start codons producing the different proteins. But there's also two other, or actually there's now another protein encoded, a helper protein encoded within those genomes in slightly different frames. So we have to take that into account when we're engineering VP3, that we might also be disrupting some of those sequences as well. Clearly, things have moved very quickly over the last couple of decades. There's many serotypes of AAV that have been explored for different reasons. They go into different cells. A lot of directed evolution and mutagenesis of those serotypes has been exploited. Display of components like scFv and DARPins on the as a fusion on the capsid has been also exploited as a way for targeting cells. More commonly, you see peptides in a loop that are now used as directed evolution targets to get them into particular cell types. People have put nanobodies on and other kind of things. And there's even some click chemistry enabled addition of MABs, etc. So there's many different ways people have started to exploit decorating and modifying the surface of AAV. Manufacturing side is in the lab typically looking like this. So often adherent cell culture in these kind of flasks, and then going into sort of benchtop systems like harvesting, maybe freeze thaw or other kind of chemical cell lysis, addition of benzonase, and then centrifuge again. But most importantly, in the lab scale, it's typically revolving around ultracentrifugation to separate out the capsids and also to separate empty and full capsid. But as good as this is, and it is actually very good at getting intact full capsids, it is not scalable largely because of the ultracentrifugation step, but also these kind of flasks aren't very scalable either. So a real process is starting to look more like this. I've shown in the bottom compared to a traditional antibody, monoclonal antibody production process. They start off similar. So the idea now is you go into a bioreactor that could still be adherent, but increasingly suspension cell culture. And then you'd use things like TFF for concentrated buffer exchange and concentration. But most importantly, replacing the ultracentrifugation is chromatography steps, typically something like an affinity chromatography followed by a polishing, ion exchange, and then buffer exchange into the final drug substance. So they're the kind of manufacturing paradigms, but there are many challenges associated with that. Primarily, the titers you get for AAV, they already were poor, really, in terms of dosing, the manufacturing capacity compared to what a typical dose would look like. And I've just sort of given an idea, if you're familiar with monoclonal antibody doses that are now hitting things like 100 to 200 mg per mil, that would be the equivalent in a dose to 10 to the power of 17 vectors per mil, which we don't achieve, right? So normally we're talking 10 to the 10 to 10 to the 15 virus particles per mil. So, we're reliant on their potency. We don't get to very, very high yield typically. So that's potentially a cost implication and a dosing implication. They're not very soluble in many conditions. Certainly as you concentrate them up, aggregation's a problem. But during manufacturing, it really is a problem. We lose a lot of material at certain steps in the manufacturing process, particularly during sterile filtration, or that TFF, what's called ultrafiltration diafiltration step. And there's lots of other manufacturing challenges associated with the analytics, which we're all very familiar with. It's very difficult to analyze all the complexities, heterogeneities of AAV. And heterogeneities, there are many. So we're all familiar with the different, perhaps stoichiometries of the capsids, different genomic variants, and the empty full ratio and partial capsids, but there are actually lots of other things going on the surface of capsids that may turn out to be important or may not. Things like phosphorylation, acetylation, methylation, glycosylation. So manufacturing is a big deal in getting kind of single. Ideally, you'd have a very pure, clean, highly potent version of the capsid, but in reality, it's a drug product is much more complex than that. We have done some work on looking at the formulation side as a way of improving the yields as you go through formulation. And one of the first bottlenecks, as I'd mentioned before, is in just simple sterile filtration, putting it through UFDF or what's called a TFF device. You may be familiar with more like the Vivaspin, so centrifugal membranes where you can concentrate your buffer exchange your AAVs. we lose a lot of material in that step, and we have done some work to improve the formulation. It was for AAV2. This is in collaboration with the company Pharmaron. We found that sucrose and low salt but high sucrose was very important for maximizing yield through the sterile filtration step. But it just highlights some of the challenges because while we can do it, we can get through and do that buffer exchange, that then limits the formulation options that we might have as a final drug product. So you can formulate, you can improve the stability of capsids by formulation to a certain degree. And this is just showing some of those examples where we found a formulation that meant we could store AAV2 reasonably high concentration for at 40 degrees for up to 3 weeks with no problem. So under that accelerated condition, you could get good stability. So it's possible. But not every AAV is going to be easily amenable to that or will have enough shelf life, because this is only 3 weeks, even at 40 degrees. So the alternative to trying to optimize the process or formulation is to engineer the capsid so that it is already more stable. And that's kind of what the main aim of this work is, alongside groups that will be targeting or improving tropism, potency, etc., for translation to the clinic. This means we can start to think about also engineering the surface to maximize its manufacturability. Directed evolution's out there, and it has been for a long time. In fact, in the enzyme world, it's been around now for 30 years. And there are many different methods available to do that. And many have been applied to AAVs, particularly error-prone PCR or DNA shuffling, and increasingly using peptides in certain loops to improve targeting. And most of this work has been designed for targeting, improving tropism, so getting into certain cells to a certain degree, improving transduction efficiency, but not much really focused on improving manufacturability. And much of this selection is done in vitro or in mice. And Francis Arnold, famous for directed evolution, once said, you get what you screen for. So if you screen in mice, then that's great if you're treating mice. But many of these, as they go into humans, fall over because they've been selected on mouse receptors. However, there's a history of protein engineering approaches beyond directed evolution, which have gone more into sort of targeting or semi-rational approaches. And that's what I'm going to draw on in the rest of this presentation. So using sequence information, structural calculations, and simulations to dive in and find where mutations are perhaps more beneficial for the kinds of properties you want to change. Some of that's already happened in some forms. So things like consensus designs are essentially that sort of approach, where you look at a multiple sequence alignment and you see your sequence that you already have and see whether at a given position, do you have the most popular or common amino acid? And if you don't, you can mutate towards it. And a couple of famous papers shown at the bottom here took that approach and actually came up with AAV2 with fairly similar looking list of mutations, although there were some slight differences between them, which were then able to modify or improve the tropism towards the nervous system. And some of them actually improve stability, and consensus design approaches are known to do that. That's been around for a long time as an approach, particularly in enzyme engineering, for example. So I'm going to focus on a few other strategies related to these. So one is structurally guided semi-rational design. So this is where you would mutate particular sites based perhaps on the flexibility. And we've done this in the past with antibodies, where we look for highly flexible regions of a protein based on things like molecular dynamics simulations, on the basis that if you mutate them, you can stabilize those dynamic loops and the whole protein becomes more stable. So you get thermostability. So with AAV, we can target surface loops and use things like consensus or approaches or Rosetta design on those sites. And this just puts together the kind of overall approach, still in the end looking at things in biophysics, this is where our main focus is, to look at whether the capsids are, when you produce them, are they stable? So we do thermal melting and heat treatment, followed by infectivity testing to see if they're still intact capsids and functional. And they may go on then into mouse studies and potentially on into other studies. So one approach we've taken is to look at surface loops and start engineering those and targeting them with different design approaches. And this is just a summary of kind of the output results in terms of the kind of things you might get. And interestingly, we've managed with this kind of approach, just throwing mutations at particular kind of loop sites, we've found that we can get improvements in transduction efficiency. We can get improvements in the overall stability of capsids through this approach. And this is just to break down where we've ended up with this approach, a few particular variants that you can see here how they've modified in terms of their, while we were going after trying to improve stability, it turned out that some of them were modifying tropism as well. And we particularly changing in cardiac muscle and skeletal muscle, an improvement in transduction, and one of them seemed to detarget the liver, although it kept the same transduction in these muscle cells. So, but it means we can inadvertently actually change some of these kind of functional features as well. So, and that kind of highlights the thing with capsid engineering is you change one feature, you can change other features at the same time. So you have to kind of navigate your way between those things. I'm not going to go through that bit, but in terms of another approach, we've looked at the kind of idea is to take multiple sequence alignments and analyze when certain residues are co-evolving. And we've used this approach in other systems before. The idea of this is that if you see from a large multiple sequence alignment that certain sites are evolving together, then they're probably important in a network sense, either structurally or functionally. And indeed, you typically find something like this where the co-evolving sites are clustered together structurally and to a certain degree in sequence, but it's mainly structurally driven. Like for example, this one in a system we did a long time ago showed an amino acid that was quite far away in sequence from the rest of the cluster, but it's actually important in forming this loop bend, which drew these bits of structure together. So it is a consistent network. So we've started to look at that in larger multiple sequence alignments of AAVs to find, are there networks that are important in maintaining structure or function, and then targeting those for mutations to see how important they really are. And the kinds of mutations we do, again, are consensus mutations or using Rosetta to design potentially altered thermostability predicting mutations. And Sirima has heroically carried out something like 100, in fact now 130 mutations, single point mutations in AAV2, and analyzed those in terms of their transduction efficiency, the yields we get in manufacture, and also stability in terms of heat challenges. And what she had done is she kind of identified initially these three networks that were co-evolving. I think two of them are quite similar. So networks 1 and 2 are both shown in red here. And network 3 was very much a separate network shown in green. And then she targeted these sites for mutations using those two approaches, as well as some others that are not in those networks as a sort of control approach. And she found from those variants, she got a lot of variability in terms of transduction efficiency on the order of magnitude of improving, 2 to 5-fold, and also in stability. So you can impact on stability and transduction efficiency quite significantly through just single point mutations. She has sort of looked at those and tried to analyze, is there any sort of relevance in where they are, what kind of residues they are, and that proved a little challenging to be generalist. So the next approach was to try and build some kind of predictive model, and we're all into machine learning these days, so that's the kind of approach she took, where she fed those variants in, and we calculated a certain set of features related to each of those mutations. So how strongly they coupled in the networks, something called Blossom 62, which is a score of how similar the residues are that you mutate from and to, how conserved the site was that was the target site for mutation, the prediction in Rosetta in terms of the mutation, in terms of change of stability, and also the location in a categorical sense location that we've set out a bunch of locations in the AAV capsid. So there were features that went into a set of models, and it turned out a support vector model gave the best prediction, and it gave something like 0.8 in terms of accuracy and 0.7 in specificity. To take that further, we could then use that model to predict all possible other single point mutations in VP3, and from that rank them in terms of whether they were likely to be good or bad, essentially. So keeping it simple to and then picking out a set of predicted mutations that could have been good. What she actually did was pick the top 11, I think it was the top 10 good, but one we already had was in there, and the bottom 5, and then some others that were not ranked at the absolute top, but predicted good, but not right at the top, and 5 kind of not the bottom, but predicted poor. So overall, about 30 new variants. And so this is just the comparison of the models, which I'll go past reasonably quickly unless you're into machine learning. But the 30 that were then remade were also then tested to see how well the model performed. And it was found that of the good category, 8 out of 10 were correctly predicted as good, whereas 2 were false positives. And on the bad side of things, there were a couple of false negatives, and the rest were predicted as being bad. So actually the model, which selected good from bad about here at this threshold, was pretty effective at finding new mutations that we didn't already have. So it was largely modeled, I should have said, on the transduction efficiency. She actually put in something that was a composite score between transduction efficiency, stability, and yield, but it's mostly transduction efficiency. And so that's really what was being predicted, I would say. Why would we do this? I guess the kind of approach here is that you don't have to make all 10,000 possible mutants to work out a machine learning model or to predict what to do next, but you only need to make about 100 and you can start to predict the remainder. Obviously, as we add more in, we can predict again and rebuild the model and make it more and more predictive. But it's a very quick way of kind of pre-filtering any library designs you might have for other purposes. So if you wanted to make a library to improve tropism or neutralizing antibody evasion, you might filter, before you build that library, filter it for any you know are going to knock out transduction efficiency, and just don't make those ones. So that could be kind of a major use of this. So you kind of defining your libraries to be more useful. This is just a breakdown of what came out, and in terms of the prediction, it showed us that the Rosetta score was reasonably discriminating between good and bad. So it was kind of one of the key factors, but probably the most important factors were the conservation score of that site and the Blossom 62. So pretty standard bioinformatics tools were quite discriminating between whether a mutation is likely to be good or not. And then the coupling score didn't have a great deal of impact, but that's largely because there actually aren't many residues in networks. So it's kind of biased out of the system. Okay, the final third strategy that we've been taking and exploring is to just use generative AI. So the idea here is with generative AI, you can now use something like protein MPNN to completely redesign a sequence into a given structure target. So if you've already got the structure, you can completely redesign the sequence. And you can do that either with the protein on its own, you can do it so it binds to a ligand, or you can do it as we're doing to form an intact capsid. Our approach that we took actually wasn't to redesign the whole protein, but we wanted to keep the function. So we left all the surface features intact, but modified all the internal residues, all the buried residues and interfaces between the subunits, and redesigned those completely. In order to do that, we had to do it first with, because the capsid is assembled as with a 5-fold axis, a 3-fold axis, and also a 2-fold axis. We had to maximize the interfaces at each of those axes. So you can imagine we built a trimer and then looked for sequences that were compatible with that. We built a pentamer, looked for sequences compatible with that, and then we built a chimera from the sequences that came out of that. And that was our design, with the intention of being able to assemble it into a complete intact capsid. And I was kind of, I suppose, only half expecting it to work well. And yeah, it worked well, it did. So in terms of normalized transduction efficiency, so we've measured how many full capsids we had. The transduction efficiency was largely, I mean, within a log order of noise, it was largely unaffected. So the function was retained, and that was probably the most surprising result, though we did, of course, keep the outer surface of the vector intact. What it meant was the vectors were assembling still, and that's the most important thing, because we're redesigning all the internal sequence. So we managed to still get functional capsids that assembled, and they retained broadly the same function in terms of, at least in terms of infecting hex cells and producing GFP. And that's with up to about 30% new sequence in the end in terms of mutations. What we did find, and this sort of goes back to a comment I mentioned earlier, was our yields of production went down to sort of, I think our best was about 10%, and some of them were down to 1 or 2%. And it basically was because we didn't try to retain the AAP embedded in the frame shifted sequence. So we knocked out, put stop codons in various places in AAP. So this is expression levels knocked down to where you'd expect when there's no AAP. And our next step actually is to try and recover that by putting AAP back in. I just want to finish on kind of where we're going. Simulations are something we like to do on other systems, and we're now producing both empty and full capsids for simulation in MD simulations and all atom simulations. So these are now ready and in progress, and yeah, I'm kind of excited to see where this will take us in terms of engineering and other areas. But yeah, if you're ever interested in simulation side, come and talk to us. So yeah, in summary, we've taken a number of different strategies where we're starting to see what parts of the sequence of the capsid affect things like stability and titers and transduction, and purity is the next thing to come out, whilst almost completely ignoring all of the usual clinical outcome things that people are after. But eventually we'll be able to marry up with people who are engineering for these purposes and create capsids that can do everything that we want. So that's kind of the challenge, is that multi-property improvement side. So with that, I think I'll just acknowledge those who were behind most of that work, and our collaborators largely in the School of Pharmacy and yeah, the TIN network, who've been very supportive around these kind of areas as well. So that is it for me, and hopefully all in good time. And thanks to you, and I'd be happy to take any questions, of course.
Thank you so much, Paul. It's really a nice and interesting presentation for us, because in the gene therapy side, when we came to manufacturing or scale up, especially clinical trials, this is really a challenge. And there is a couple of questions, and I want to forward to you. The first question is that, how will the new AAV2 mutation affect manufacturing?
So the idea is that we are actually assessing the manufacturing. So if we get higher yields, so that's kind of where that work here was going. So these are producing higher yielding capsids. So this is, which one is yield? I can't see my own screen. T vector genome yield here. So we did have one, this is the first round, we had one variant which jumped out above wild types down here with the dotted red line. So this is a yield of something like 14 times in terms of production. Okay, so that's kind of rare to get that big a jump with a single mutation, but that's the kind of thing we're looking at here. And if they happen to improve stability as well, then potentially they will survive better during the manufacturing process and TFF steps. We are currently starting to put these into things like TFF analysis and chromatography to see how well or how much they have impact on yields going through those steps as well. So that's kind of ongoing.
Also, I want to ask another question related to this one. For example, your main focus is stability, but after create a mutation to be more stable, possibly there is another effect, unknown effect can happen, maybe another target, like how you can estimate it, this feature of this mutation.
Yeah, I mean, unless you actually measure it, you don't know. And that is something we're finding. So some of the earlier variants I was showing you, we have put into mice. We've started doing human cell testing as well, and some of them have shown surprising improvements in essentially the transduction efficiency into different cells, or the tropisms change. So you can kind of see that it's very difficult to decouple, right? So at this point, we're not at the point where we have like the magic mutations you can apply to any vector without affecting things like the tropism and potency, but we are starting to learn which of the palette of stabilizing mutations affect which kinds of tropism, and I think that's the way it will come out in the end. You won't be able to sort of isolate the surface mutations as having only one effect. They're always going to have multiple effects.
Yeah. And next question is, how confident are we that the new AAV capsid made by AI tools will be suitable for manufacturing? Like, okay, we expect they had to scale up, but how much times are still doing it to some in vitro or in vivo validation?
Yeah, it's way too early to be thinking about the AI designs in anything clinical, etc. As I say, they retain their function based on the measures, in simple measures, transduction efficiency of hex cells with GFP. So it's not a clinical payload. It's not really in a very clinically relevant cell line. It is at least a human cell line, but this isn't looking yet at anything about where it's going in terms of biodistribution. So it's a long way off. This is really kind of winding back to, can we engineer these using Gen AI at all? And the answer is yes. I think that's the first step, is that we can actually get functional vectors. Next step is to start asking that question: can we actually use it to sort of scale the mountains we want to? At the moment, it's not back the yield so far that it's actually made manufacturing worse. So we have to recover that in the obvious way we think we can. But eventually, that's if we can get this back on a par, then we can start talking about redesigning for manufacturing.
Yeah. And I think there is some safety concern question about the new capsids, but I think you have already mentioned that, like, explain the process already. But if you want to add extra, please go ahead. Or
Sorry, in terms of what I kind of missed
safety concerns of the new capsid.
Oh, of this one. So we were very careful with this. Obviously, just kind of flying out and mutating potentially could create a whole new viral vector. But no, we're very careful with this. Mainly, it's still got all the safety features in that it's non-replicable. It doesn't have RepCap as its payload, so it can't survive in the wild, of course. We were also extremely careful to make sure it was really the same, right? It's got the same function because it's got the same surface as the wild type vector. So really all we're doing is it impacting the internal workings of the protein folding. So for this one at this point, it's no different to creating stabilizing mutations of existing serotypes. We're not changing the functional properties.
Yeah, thank you. I think another question is, are the new generated AAV capsid designed to target certain tissue and organ?
That's not something that I focus on specifically. We are seeing that certain surface engineered vectors that we make do change those kind of properties. It was never the design, but it's almost an inevitability of doing surface mutations. So you do have to track that, and the more we track them, the more we understand them. And some of them are fortuitous changes, so you kind of go run with them, right? Yeah, it's not what I do, but certainly colleagues here at UCL, that's their business, right? Getting into particular cells for disease treatment purposes.
Yeah, I agree. And these are all questions, Paul. Thank you so much again for your explanation and bring the new perspective for gene and cell therapist. Thank you so much for that.
It's a pleasure. And thank you for letting me give this presentation.
Our pleasure. Okay, we are